Hi guys, my name is Derek and I'll be running through some ideas, concepts and thoughts behind the lab report that you might be covering. So, looking at what I've got, this is some key ideas of what you would be expected to write, such as your abstract, your introduction, methods, results, filling out this table, which you will do in your labs. This graph that you need to plot out, and this table again, and some points to consider here. Alright, so maybe let's start from the top where we can explain what we're trying to get across. So this experiment, as said here, is to introduce red blood cells and maybe put them into a bit more salty fluids. But to break that down and to get some ideas across of it, this experiment isn't even really talking about red blood cells, but red blood cells are just the most convenient membrane in biology to watch and fail. They're free, they're everywhere. They don't really have a nucleus or organelles to make things complicated, so you can just focus in on that membrane. Because once the membrane breaks, you can see it with your naked eye. The tube goes from this cloudy substance into something that's clear. So what we're looking at specifically, and what this experiment probably is going to be about, is going to be this plasma membrane. So the idea is that blood sits in fluid, and its survivability, the blood cell survivability, depends on the balance of the dissolved fluids in your body and within the red blood cell. And so we're trying to understand, with changing these concentrations of the environmental salinity, or how salty the water is, or other liquids, how does this affect the red blood cells' membranes? So we're getting more than this lab result, and we're getting something more useful to understand with the results that we interpret from this lab. Hi again. Now, following that small explanation, once we've gone through in the lab and done the experiment, you move on with filling out this table, and once this table is filled out, you complete this graph, which would look something along the lines of this. It's alright if it doesn't follow this shape, and if it doesn't follow this shape, we can just write more about it in our discussions inside this lab report. How we can get the results and interpret it and plot it on the graph. We can get into the nitty-gritty once you cover the lab, and once you have your results, and we can work through it together maybe in the near future. But with that being said, after getting the results, you might want to move on to writing the report. And here is where I say to write in this general order, or to think in this general order when writing it. First of all, you start with the methods, which is the easiest because you just did it. And then write about the results, because the data is already done with our calculations and us plotting on the graph. Then, since we got the data, we can maybe start on the introduction now, where now you know what it needs to set up. And then write about the discussion, where you discuss and interpret the results. And after that, write about the abstract. Last, because we just summarize everything we have written, because what an abstract is a miniature whole report which is like readable alone.